What up, everybody? How's everybody going? Welcome to another episode of Whose Gene Is It Anyway, where the ethics are made up and the species are made up and everything's made up and it's great. Um, Either way, welcome to the show. Um, today's gonna be a fun one, I think. We're gonna do so the just to catch people up who missed the last one. The basic idea of this stream is throughout the course of the live stream, I am going to first pick an idea out of a hat. Here is the hat. Note my lovely plague doctor mask full of scrunched up ideas. So I'm going to pick a random idea out of a hat. Many of these ideas were submitted by you. So I think this is going to be. Some of them are pretty wild. I'm a little nervous. I kind of hope that I get an easy one. There's some hard ones in there that I think are probably going to kick my ass, but I think it'll be a good way to practice and a good way to, kind of force myself out of my comfort zone, right? So I think that'll be fun. If you'd like to submit ideas, send them to me on Twitter with the hashtag Who's Gene. I don't know. I never do Twitter hashtags. Let's see if that works. So yeah, I think we should probably just kind of get right to it. So the first thing I'm going to do is I'm going to draw an idea. And then, as I was saying, throughout the course of the stream, I have to research the idea and then build a piece of DNA that would accomplish that idea. So I think this is going to be a lot of fun. I'm going to draw the idea, and then we're going to quickly talk about the things that we made last time. And yeah, so this is going to be fun. There was a quick question. Did you qualify the ideas before going into the hat? Yes. I'm I'm not that much of a masochist. I made sure that any of the ideas that went into the hat seem plausible. Like I did a quick 2 minutes worth of research just to be like, okay, am I about to look like an asshole on stream, or is this actually doable? For example, chocolate milk cows. Sounds easy, is not. Um, can't do chocolate. Chocolate's too hard. So yeah, this is gonna be fun. Anyway, let's go ahead and draw an idea. Scrunchy. I'm just gonna grab a random one. I got a couple. Okay, this one. All right, wish me luck. Let's see if this is good. Oh, it's a yeast one, so that's exciting. Oh Lord. Oh no, this is gonna be hard. So hopefully you can read this. It says yeast that produces an antibiotic. We'll see. I think what I'm gonna end up doing is we'll do a little bit of research and we'll see how viable this is, and if within 10 minutes it doesn't look like it's even possible, then I'll draw another idea. So. Okay then, this is gonna be fun. Okay, let's just get at it then, because this is gonna be exciting. Okie dokie. So things to do. Before we get going, this is the this is the stuff that we made last time. So for those unfamiliar, this program is called Genome Compiler, and we're going to talk about Genome Compiler in a second. And these two circly things are basically like the programs that we wrote last time. So there are two here, and they each do a different thing. The first one being this one. So the theme last time was milk and eggs. It was, could you make milk and eggs without animals? And so what we did was we designed these two pieces of DNA. This one on the left it's built to work in yeast. So you would take regular brewer's yeast or baker's yeast, whatever, and you would put this little piece of DNA into them. And when you do this, it gives the yeast some new abilities. Thing number one, it makes it resistant to this antibiotic, which is it's labeled kanamycin, but this actually gives them resistance to an antibiotic called G418, which is just like a modified version of kanamycin. And then this is this sort of interesting bit where it would produce 4 different proteins that represent the major components of milk. So 2 different kinds of casein and an alpha lactobulmin and then a beta lactoglobulin. So in theory, if you were to put all of these, so if you put this into yeast, it should make these 4 proteins, and the brewing liquid would basically turn white. That's the plan. And then the other one was we can't do egg yolk because the proteins in egg yolk are just too large, and so it's not feasible. So instead we did egg white, and so the main component, about 50% of the makeup of egg white is called ovalbumin or albumin, and so all we did was we just kind of put it in the same empty yeast plasmid, and we just stuck the ovalbumin gene in there. And what all of these proteins have in common is we've stuck a little secretion tag into the front half, which basically just makes sure that any proteins that are produced end up outside of the yeast and don't stay like trapped inside. So this way, like if you were brewing the egg white one, it should turn into a tank of egg white. And so now you may notice that there's a donations bar now. So basically, I think these are really cool. There was a lot of excitement about these plasmids, and I explicitly designed them to actually be synthesizable. So like I could pay a company and they would just make them for me. And I've got a company that I like using for this. And so I'm thinking that I've got the donation bar there, and I estimate that it's probably going to cost around $1,400, give or take. Could be a little less, hopefully not more, to get both of these made. So with the donation bar, there's a link in the description for those who would like to donate. And yeah, so if you donate, and we can raise a little chunk of money, it would make my life a lot easier so that I can pay to have the synthesis done. And then these plasmids will be first I'm going to test them, make sure they work. And then once I know that they're working, they'll be submitted to a company called Adgene, so that anybody who wants them can just buy them. And Adgene will just like send you a little vial of bacteria with this DNA in it. So then you can take it and do whatever you want with it. I'm also thinking about maybe setting up a store on my site. I haven't decided yet. And to that end, to people who donate, if I do end up doing the store, I'll keep a record of everybody who donates, and that way I'll give like a discount or free shipping or something. Like I'm not going to be charging like huge amounts of money for the plasmid. It'd be like the cost of the little vial, the agar, and my time to like, set them up. But like, basically, I'm going to be like selling them at cost with shipping is the plan. So yeah, if you donate, give you a basically free. So like donations over, let's say $15, free shipping, and I'll keep a record of everybody who does, and then we'll be able to get the thing made. Hopefully, this is something you are interested in. Yeah, I think this will be a lot of fun. Fairpoint Station. How much to analyze my stool sample? More than you have. I'm not that kind of biologist. Okie dokie. So, I think this is gonna be a lot of fun. So these were the ones that we made last time. As things go, depending on how the fundraisery thing goes, like this could be something that we do regularly. So, because I'm intending to do these streams at least once, if not twice a week. So in the end, we're gonna have made a lot of plasmids. All of the plasmids that do get designed, I've been uploading to this lovely GitHub repo. So if you want to play with the plasmids that I've already made or that get made on the stream, this is the place for it. I'll put some links in the description underneath underneath the description of the video. So if you want to get to this, but if you just look up whose gene is it anyway, and I realize I'm an idiot and I misspelled this, I'll fix that later. But yeah, so anyway, somebody just kicked in $5. Where's my list? Why can I not see who this person is? That's disappointing. Well, whoever just pitched in $5, thank you very much. Okie dokie. So with that out of the way, we've there's one other piece of news that I need to address, because I literally found this out the day after I did the last stream, and this kind of broke my heart a little bit. So Genome Compiler, this program here, I've been using for a long time. I really like it. It's very easy to use, and it's clean, and the colors are nice and bright, which I realize this has essentially no bearing on, the usability, but like when you've got to stare at something for a long time, having like a nice color palette is kind of good. Yeah, turns out if you go over to the login page, cool. It's just gonna log me in directly. That's fun. Okay, well let's close this. Yes, leave. Thank you. So let's just go to the login, and hopefully it doesn't. No, it just. Okay, I've got to actually log out. That's annoying. Basically, I want the long and the short of this is, this is what I wanted to show you. Access to Genome Compiler website will be disabled on 9/30/2020. Users will still blah 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 blah. Point is, Genome Compiler is shutting down, and this kills me a little on the inside, because it's such a wonderful tool. I think what happened was they got bought or sold, the company or whatever, to Twist Bioscience, which is a gene synthesis company, and presumably they're going to be integrating their tools into Twist's, but shrug, I don't know, man. I'm not into it, and it means Genome Compiler is shutting down, and this is very shitty, and I don't like it. So, on top of having to design yeast that produces antibiotics, I'm going to have to do it in a new tool today. So, let me introduce you to... Ooh, thank you. Thank you, Dr. Good Doctor. Allow me to introduce you to Benchling. Benchling very recently added a whole bunch of tools that essentially do the exact same thing as Genome Compiler. To the point of it looks like they basically cloned it out with a shittier color scheme. Benchling normally is used for taking notes. So one of the really good things is, so we're in Notebook right now. So it's really great for like writing out protocols. And I've used this for many years for doing this thing. So it lets you write out your like bill of materials and whatever your procedure is. And I've used this for a very long time. It's very effective. It's also nice because when you're like designing a thing, you normally are like taking notes, because I do things a little differently, wherein if I can avoid having to do actual lab work in the, like, when it comes to putting DNA together, I will. And so I design things as if they're going to be synthesized, fully complete, no need to mess with them, just ready to go. But assuming you're doing something where you actually have to mess with the DNA a little bit on the bench. Oh, somebody. We just got a donation that says, I guess solving world hunger is fine, I guess, but I'll be waiting for open source cat girls. Well, I mean, cat girls are hard, and also, wow, does that cross ethical line so quickly. Well, anyways, thank you for the people who pitched in. Y'all are great. Anyways, back to Benchling. So I've been using Benchling for a long time. If you're designing something where you're going to actually have to do some bench work, like say you need to like PCR out a fragment, stick it into a new plasmid, whatever the case may be, you always take notes, right? Like you're taking notes. You're like, okay, I'm going to need to cut with this enzyme, and then I'm going to have to do this process, and just so you know. Because there's nothing worse than getting a very expensive piece of DNA in the mail and then completely forgetting how you're supposed to handle it. So that's exciting. Okay, so I'm just going to answer a couple quick questions. Have you looked into sanitization with UV light? I mean, yeah, I use UV to sanitize stuff all the time. You just got to get the right kind, and the kind that actually sanitizes things is not the kind you want to be around when it's on. Um, so yeah, that's a thing. Okie dokie. Oh damn. Well, thank you, Todd Gore. You awesome. Um, anyway, so let's actually. So now I've kind of given the introduction. Let's get some work done. Let's do some things. So we need an antibiotic. I think penicillin. I don't know. Let's just start with penicillin. Biosynthesis. Let's see how penicillin works. PMC. What's data nexus? What is that? Before I look it up. So I think the only donations that go to the bar are the ones that go through the actual donation link in the, in the link in the description of the video. I believe super chats do not. I'm also keeping a note of the super chats. So, yeah, just if you want to move that bar forward, do the donation link. If you want to just like pitch in because you like the show, super chats is also a good option. But again, I'm gonna keep notes of everybody. So assuming that I do get a store set up, we'll just make it so that there's some way to like provide the username, so that I can check. And apply that discount or whatever. Okie dokie. Let's learn how penicillin is made, and if this pathway is. And so this is something that I actually do a lot. Biology is inherently physical. Ooh, data nexus for the win. Okay, now I gotta scroll up and find that thing again. Okay, so whenever I'm doing research like this, I like to see how these things work. Because like biology, even though you're doing stuff on the computer, like you're dealing with inherently physical things. So it's really important to see how these things come together. Because it's really biology is just aggressively complicated chemistry. Here, let me just. I want to see if I can find the thing just to illustrate how ridiculous this is. Yeah, okay, here we go. So here is a chart of, assuming it'll actually open. Come on. Of course, it's always like, wow, shit. Would you just open, please? Cooperate, goddammit. Really? Okay, well, that's frustrating. The thing I was trying to show is that like the metabolism and like just the way that compounds get shoved together and mixed and matched in biology is ridiculous. So yeah, things can get complicated. So it's really important to actually like look at the chemistry and see how it works. So in this case, here's a synthesis for penicillin. Ooh, some more donation-y goodness. Okay, so I'm going to just copy in this thing, PMC 58595. Okay, let's see what this is. Biosynthesis of non. Oh, here we go. Wow, that's really handy. Wow, this is basically describing exactly the thing that we need to do, which is super handy. Okay, so let's see. Yeah, I think this is actually exactly the synthesis pathway that I was looking at. So by the looks of it, there's one, two, three, four, five genes, I think. I think it's a 5 gene pathway, which, ow, but not so bad. Now the real question though is, will these people actually include the genes? So I always like to jump to okay, let's just look at their methods really quickly, see what's up. Um, okay, just checking the comments, see what's going on. Ooh, we got more people pitching in. Thank you guys. Y'all are awesome. Guys, gals, whatever else. Okay, let me just checking some stuff. Making sure that everything is hunky dory. Yeah, okay, all looks good. Oh, yes. Thank you for the reminder. I need to change the stream title, so I'm going to just do that really quick. So we're doing penicillin yeast. There we go. Save. Okie dokie. The stream title has now been changed. So now people know what we're working on. Okie dokie. So now we're actually let's read this paper, see what's going on. PST to this, okay. Native C-terminal tripeptide. Interesting. So a very quick review of their methods section mentions the peroxisome. So peroxisome is an organelle in eukaryotic cells. It's sort of what it sounds like. It's basically a little bubble that fills with peroxide. And so seemingly that's where the penicillin needs to be made, which is interesting. Neat. Oh, I don't have the chat open. You may need to refresh the stream to see the title change. Okay. So yeah, in theory, this should be fairly straightforward. By the looks of it, just by how they're naming the genes, is they've probably stuck several of the proteins together into what's called a fusion. So it's literally just like two proteins stuck together. And as long as the active site isn't inhibited, so there's like a spot on an enzyme where the actual chemistry takes place, and as long as that isn't messed with and the actual like structure of the enzyme is unchanged, you can actually stick all sorts of different proteins together and make like one big super protein. Interesting. So yeah, they're doing this just as I'm reading this. So they used Golden Gate assembly, which is not how I would ever do it because it's just tedious. It's not even tedious. It's mostly just expensive because you need a bazillion different enzymes just to match and do the thing. How do you do protein fusion? Well, you do a little dance and then you do the thing. No, it's you basically there's a it's called like a linker peptide. It's basically a string of glycine, so like 5 glycine in a row, that you just stick between the two sequences, and then they'll just form stuck together. Do you recommend buying a nanopore if you have enormous amounts of money? Sure. Nanopores are great. They're just very expensive. Um, and like, unless you need them, I don't know why you would. Okay. Oh, hello. Plasmid construction sequences are provided in supplementary data 1. That is exactly what I like to hear. There's... there... Ooh, we got another donation. Thank you. There's nothing I like to hear more than we have included the supplementary data. Plasmids, hello. Dude, this is fantastic. Oh, this is great. Okay, well, so let's load this into okay, come on. There we go. Cool. Well, let's load all this into Benchling. Let's make a new folder first. New folder. Wait, no, cancel. Got to do an inventory. So this is the one thing with Benchling. It separates genetic stuff and note stuff into two different categories, and I didn't know this when I first tried to use this, and it was kind of obnoxious because I couldn't find any of my stuff. Anyways, let's get this. So we're gonna call this penicillin yeast. It's kind of weird designing a thing that would maybe kill me, because I'm allergic to penicillin, or at least I was at some point. I don't know if I still am, but I'm not willing to find out. Okie dokie. Let's import some DNA sequences, and let's open up all the stuff that I just downloaded. So plasmids, there's a huge list of them. Hopefully they're annotated, because otherwise that would be immensely annoying. Okie dokie, they're all in. So let's have a look and let's see how they. What? Is this just done? Ooh, gross. Uracil. Oh. So the thing that I just kind of like barfed at a little bit is that they use actually no, it's just this uracil promoter. Oh no, that's the actual. They're both there. So yeah, the way that they select the yeast that have been modified once they actually put this DNA into them is they grow them on media that is missing uracil, which is one of the amino acids. In the same way that we have amino acids that we can't survive if we don't eat, yeast have amino acids that they can't survive if they don't eat. Like, they just don't produce them internally. So one way that you can select for the modified colonies is you basically grow them on media that is missing that one amino acid, and then you give them the code that lets them synthesize it themselves. Is any of this annotated? More importantly, do the annotations show up? Description. No. Metadata? Also no. Okay, well, rip. So penicillin doesn't actually work on fungi. So there's only the whole reason that I use G418 selection on my yeast is because most antibiotics don't actually affect yeast. Like, the way that they function is that they actually inhibit or break or mess with processes in bacteria, whereas yeast, being eukaryotic, behave very differently. So you can grow yeast on just enormous concentrations of kanamycin, and they don't respond to it at all. Whereas if you tried to grow like E. coli on that, it would be dead instantly. And yeah, a lot of antibiotics are created by fungi. A lot of bacteria produce antibiotics as well. In this case, penicillin comes from a fungus. So the chances of it actually affecting a fungus is very, very low. Annotated gene bank files. Yeah, okay. So I don't know what this 2-micron thing is, but just from the little bit that I recognize from the paper, this looks like basically a completed plasmid. I want to see what these other ones are. Coding sequence, coding sequence, coding sequence. Why is this one smaller? I'm going to just open all these up. We're going to just see what they look like. Okay, here we go. So, wait, what? This is very strange. So it looks like they've got multiple different constructs, and they've kind of mixed and matched to sort of end up with something that functions. So I'm not sure which one is the actual functional bit, because they all look kind of the same to me. All module. Yeah, I don't know what this is. Okay, so 180 I don't think is useful to us. So let's pop back over to the paper. Stronger expression. Okay. So let's just read through this really quick. So yeah, this paper claims that this is the first biosynthesis of bioactive antibiotic using engineered yeast, which is cool. Expression. So it seems like they've made benzyl penicillin, and I don't know what the difference between that is. Let's find out. What is the difference between benzyl penicillin and regular penicillin? Or are they the same thing? Oh, they're exactly the same thing. Okay, so penicillin is benzyl penicillin. That is the actual proper name for it. So presumably penicillin is just the other name. Penicillin G, penicillin sodium. Yeah, that's penicillin. That's okie dokie. So they do actually make penicillin, and so this paper also talks about optimizing how to make it work the best. So in this case, it looks like says we observed that this required strong expression of the peroxisomally located enzyme PCLA. Which I don't. Let's find out what this enzyme does, because I don't know what it is, and I'd really like to know, and it sounds important. Yeah, okay, so this is from ligase, penicillium, calcium. I mean, it looks like it's from the Penicillium species. But, I mean, beyond that, it's not really telling me what it does. Okay. We reasoned that cytoplasmic expression of all enzymes would not suffice. Interesting. Okay, so PCLA and Pen D and E seem to be the things that actually make the penicillin. So actually, let's just scroll up and it should just say. Yeah, here we go. So PCLA is part of the penicillin synthesis pathway. So if I'm understanding this correctly, it takes PCB A and B and NPG-A to go from basically a collection of amino acids into something called ACV, which is presumably a precursor, which if I had to guess, was adipitic acid, cysteine, and valine stuck together. So it's basically, this is, other than the adipitic acid, this basically looks like a short peptide, essentially. And then the PCB-C seems to grab one of the dangly bits, the thiol group. It's hard to see. I'll see if I can zoom in. Yes, I agree. Dismiss. Yeah, so there's a thiol group and an oxygen. It seems to just, does it shove that together? Or does it? No, it takes the thiol and it sticks it to the nitrogen in the back here. Interesting. Okay, so that gets you isopenicillin N, and then you need 2 more enzymes, one of which looks like a fusion, to actually get the benzylpenicillin. The dangly bits are back. There's always dangly bits. So it looks like it's hard to tell if they're using 3 genes or if they're using separate plasmids all stuck together. Okay, strain SCA1 produces ACV. Okay, so it is 2 plasmids, I think. One gets you, so yeah, one of them gets you the ACV precursor, and then the second one does the little flipperoo to turn it into the next thing. Does that look right? Yeah, because they've got PCB-C, PCLA, and PEN-DE. So that's PCB-C, PEN-CLA, and PEN-DE. So these 3 seem to be on one plasmid, and then they've got the PCB-AB and NPGA on a separate plasmid. And so I don't see any reason why you couldn't just shove all of those into one thing. It seems some of these need to be in the peroxisome, presumably the second half. And we should see all that reflected. So there's a question: how are you going to get the filamentous fungus that the gene is taken from? I'm not. When I design DNA like this, I assume that everything will be synthesized chemically. So unless there's some reason why the gene can't be synthesized, like for example, it is extremely repetitive, like is the case with most structural proteins, then you basically can just synthesize the entire thing all in one shot. You never need to mess with the source organism. You never need to mess with PCRs or extractions or any of that. You just like tell the company, make me this gene, put it here in this way, in this plasmid, and they'll just do it. Yeah, just to recap, I'm making a yeast that produces penicillin. So this is just for those who want to follow along, this is the name of the paper. Biosynthesis of antibiotic non-ribosomal peptide penicillin in baker's yeast. And I realize now that I'm starting to learn how this works. I really should have caught on. Like I noticed the peptide thing and I was like thinking about that, but it really didn't click just how peptide-y this was until I saw this and I'm like, oh, it's literally like 3 amino acids stuck together. So what's this figure look like? Okay, this is just growing stuff. Penicillin. And by the looks of it, it says yeast culture supernatant. So the penicillin leaks out of the yeast and just into the supernatant. This would be pretty cool. I really don't think I would ever actually have this made, but it's pretty neat. Like it's a really neat thing. Like if you needed just a tank of penicillin, this is a good way. You know, it's at Patreon, Ko-fi. Like there's lots of ways. Sometimes I do sponsors too. This is my job. I'm entertaining for a living. At least I hope I am. But yeah, it is how it is. It's a fun job. Albeit a little demanding at times, but considering that I'm one of few people who continue to have a job in a pandemic, I can't really complain. Okie dokie. So let me just. Interesting. Is it lagging? Yeah. Wonderful shit to your internet. Well, that's fine. Okay, so let's open up Benchling now that I kind of understand what's going on. So yeah, now that I know what's up, I can look at this and go, okay, this makes sense. So yeah, we've got PCB-C, PCLA, and Pen-D. So this plasmid does the actual like flipperoo, turn that little peptide thing into the actual penicillin. So that's cool. So 179 is make penicillin from precursor. Somebody pitched in. Very exciting. Thank you. Okay, yeah. So this also looks like it would just make. Oh, actually, no. This one has, I think, all of them in one shot. I'm seeing PEN-DE, PCLA, PCB, C, NPGA. So this is missing one, I think. This is what's this missing again? Where's my chart? Yeah. So this is missing PCB AB. So. Man, this might actually be way easier than I thought it. Well, I mean, it's easier in the sense that they actually aren't just the worst, and they actually provide all the plasmids. Oh, there it is. How large is that? Oh, wow. Okay, that's a chunky boy. Okay, well, shit. So yeah, there goes any hope of actually just having this synthesized, because, yeah. That's okay. I'm gonna close this because yeah, no, I mean they're all open. Right? I should just be able to. What? No, Jesus, close that. There we go. That's what I wanted. Okay, so I can zoom in on this a little bit just so I can actually see all my stuff. Great. Yeah. So this is so just seeing this is the reason why. Somebody kicked in. Thank you very much. So yeah, just seeing this one gene instantly tells me that yeah, you're not. I mean, you could have it synthesized. It would just cost you an arm and a leg and a dick. Synthesis is expensive. Like it saves you. It ends up actually saving you money because you're not burning money on supplies and kits and time. Like your time is worth money. So like if you've got to burn 6 weeks sitting at a bench burning through materials trying to get a reaction to work, like you're pissing away money. Whereas if you just have something synthesized, you might spend a little more up front, but you're saving yourself time and money later. Seeing something like this where length of 11,331 would be very expensive. Like very, very, very expensive. You'd probably have to have this made in 2 or 3 pieces and then stick it all together. And you can pay the company to do all of that for you. So you could break this up into 3 parts or 4 parts, whatever. Have them synthesize the individual pieces. Have them put the pieces together. Put that big piece into the plasmid that you want. But what's going to end up happening is it's going to cost you a lot of money. That's basically it. So like this whole to actually make this whole thing. Rather, because I mean, one of the other options with this, in the same sort of sense that like you can email people to get a copy of the software they wrote, you can email the people who wrote this paper, and I don't know if they'd actually give it to you. They might because they've published the actual plasmids and stuff, which, from my perspective, makes me think that they're kind of interested in sort of open sourcing this. Because when you publish a paper and you include this data, it means that anybody can actually just copy it and get it working themselves. So to me, this makes me think that maybe they would be open to, like, if you sent them an email and asked really nicely, and I don't suggest that everybody do this, but like in general, if you see a paper that they've made something really cool, a lot of the time, if you're just polite and ask really nicely and professional, and you can just email the people and be like, hey, this is a really cool thing that you made. This is what I'm working on. Would you mind sending me either the modified yeast or just the plasmids or whatever? And a lot of the time, people are just happy to give you stuff. Like they just appreciate, um, like that you care. Because I mean, a lot of the time, people will spend years and years developing something like this, and it just never goes anywhere. Like it's just they make it, and then they write the paper, and then they move on, and it just sits in a fridge. Um, so most of the time, they're actually pretty good about just like sharing stuff. Ooh, we got some more. We got some more donations. Thank you very much. Um, so yeah, just because this would be obnoxious to synthesize, honestly, the best first step if you actually wanted to do this would be to email the authors and be like, "Hi, um, this is a really cool thing. I would like to use it for this purpose. Would you mind sharing?" Basically, and do so in a professional manner. But because that is not the point of our stream, like that's how I would do this if I wanted to actually do this. But that is not the point of today's stream. The point of today's stream is to put together a plasmid that actually does the thing. And the nice part about this is, oh man, they've got kanamycin resistance. Man, this would be such a great plasmid. So yeah, the nice thing about this is, in theory, like you could just get these plasmids, but that is not the point of today's stream. The point of today's stream is to actually make something viable. So I'm gonna go back to Spiderbrick main. I'm gonna go to milk and eggs, and I'm gonna grab my favorite empty yeast backbone, which was found by, I believe, Sassy Tripper Sniper. I remember them as Sassy, who is the bomb. Holy shit! Did somebody just? Holy crap! Somebody just donated $600. Thank you. Holy crap! That is amazing. Okay. Well, I'm a little speechless and floored. Thank you very much. That is incredible. I wanna actually find I need to find the name of this person and thank them, because that's just an absurd amount to pitch in, and I really, really, truly appreciate it. Like this is I I really think by the end of this we're getting these plasmids made, because this is just freaking fantastic. Who just? Oh, Nico Schluter, dude, you are the bomb, man. Thank you so much. Okay, and he included a question, which I am definitely taking time to answer. So, how can a mechanical, electrical, software engineer that doesn't really get biology best contribute? Any open source projects I should look at? I mean, you just kicked in $600 to get some plasmids made. Like, there's sort of a bit of a misconception that like everybody needs to be able to do the thing that I'm doing, which is like designing the plasmids and doing the bench work. But honestly, it's just as valuable to find people who are already doing those things and just find ways to support them. Because we don't actually need 10,000 people designing plasmids. Like, if we had 10 people making plasmids and then 1,000 people like chucking in a few dollars to help pay for the various costs, like that'd be huge. Or like, if you're, say, good at doing tax stuff. Like a lot of the time the people doing bench work don't have the time to stop and like, I mean they're kind of forced to, but like if like having to take the time to like stop and deal with administrative stuff slows things down. Whereas if those are your skills, you don't need to learn biology to help with administrative stuff. You can just help with the administrative stuff. So yeah, that's honestly just find people who are already doing the thing and see how they can use your skill set. And it doesn't need to be bio. Like genetic engineering is awesome, but it's very expensive. Like very, very, very expensive. Like bio is, and I hate to say this, but it is the way that it is. It's pay to play. Every action that you take in a biology lab inherently is pay to play. Like it's say you want to make so look, these are all restriction enzymes, right? So they're enzymes that if you mix this DNA with this enzyme, it will cut right here. Um, Thing is, each one of these costs minimum $100. Well, like 50 to 100 for something that's common, but for something weird like I don't know, this I've never heard of this one before. It'd probably cost you five, just because if it's not common, they don't produce a lot of it, so they charge a premium to get it. Um, and so like every action can cost anywhere from 50 to $500. Um, oh, thank you, Joey. Um, so it's just kind of like it is the way that it is. So like just providing monetary support for the people who do this kind of stuff is like a world changer. Like it makes it possible to actually do these things. Oh, thank you, Grumpy Lion. See, I've actually got the I'm still, this is, I mean, only my second stream, so like I'm still learning the ish of like how to how to go about this. So yeah, I actually have my like donations thing. I can thank everybody properly. Um, in fact, I want to just like go through really quick, and for just thank the people that I missed. Um, so yeah, I'm gonna just do that. Yes, and I know I need to update Chrome. I'm aware. I meant to do it before the stream, but I'm a turnip, so I forgot because my priority this time was make sure the stream was not in potato quality, and I think I managed that. So, one thing at a time. um, so yeah, there was a few more questions from some other people who donated. Oh, thank you, Valaway. So programmers interested in bioinformatics would love to get in contact. Okay, well, I'll send you guys a message after. Well, thank you very much for your donations. Um, Somebody whose username is milk and eggs, please, donated. Well, I will do what I can. Um, there's a thing, harvesting energy from ambient humidity. I've seen that paper, and it's meh. The amount of like energy they get out is minuscule to the point of it not actually being really feasible. Any yeah, so I mean, again, thank you to everybody who's donated so far. Like, this is making my day, and especially thank you to Nico. You the bomb. So yeah, I'm once we've hit the donation goal, I'm hoping that I'll be able to put in the order. Oh, thank you, Aaron. I'll be able to put in the order for I'm going to start with the egg plasmid because it's going to be the cheapest, and I'm going to also put in I'm going to get quote requests for both of the plasmids, and we're going to try and make sure that I get them both made. Anything that isn't covered by this round of donations, I'll cover myself. That way, we've all got some skin in the game. And yeah, hopefully we'll get it done pretty quickly. The company that I use, who again I'm not gonna give the name of the company that I use because they're awesome and I don't want to send a huge flood of traffic their way and like piss them off. But there's lots of different there's lots and lots of different places that will do gene synthesis for you. There's Twist, there's IDT, there's GeneScript. I don't use any of them. I use a different one, but they all do the same. They all do the same job. Yeah. So I'm gonna put that quote request in. I mean, assuming we hit our donation goal, which at this rate we might. I'll get that quote in probably today, and then I'll figure out how much this is going to actually cost, and then I'll get them made. I'm going to actually have to go in and make a few changes that I realized that I forgot to do in the last stream. So, I want to... I'll just open it up so I can show you. Gotta say, I'm liking how fast benchling is compared to Genome Compiler. Like, it definitely doesn't look as pretty, but comparatively, it just... everything's, like, quick and smooth, and I do like that. So yeah, this is the thing that we made last time. So the one thing I want to do is I want to add specific restriction sites between the various proteins so we could cut them out if we wanted. I also need to add a stop codon to the end. But yeah, that'll be for another time. So just small little changes. Then I'll put in the quote request. Oh, thank you, Sebimo. We're going with Sabimo. Thank you. And with the question, how do antibiotics actually work? That depends on the antibiotic. Some of them cause the microbe to literally lyse, like it attacks the cell wall and pops them open. Some of them are, um, some of them are like transport inhibitors, so they'll mess with the cell's ability to like either bring in various like things that it needs, or it'll force it to like dehydrate, like it can like grab onto the like one of the export proteins and force it to just like leak water until it just explodes. Some will go in, like tetracycline messes with protein synthesis, for example. So it binds to the ribosome, I believe. I'm going off memory, so this could be totally wrong. But I believe it messes with the ribosome and prevents it from actually functioning. So no protein synthesis, and the thing just dies. So a question about the milk and eggs. So one other thing that I'm going to add to the milk and egg thing is what are called LOX-P sites. So basically these are integration sites. So basically what'll happen is once you put the DNA into the yeast and you use the antibiotic to select for them, what will just sort of happen randomly are integration, so like basically what'll happen is the bits of DNA that are flanked by these integration tags will essentially get shoved into the yeast's actual genome, making it a more permanent modification. At this point, you can then switch from using the antibiotic as your selection agent to you can just grow the yeast as is, and then it should just keep the mod. So yeah, you can just like grow it in bulk. So like I just off screen, I have a flask of yeast that I modified to produce beta carotene. And those all come with integration sites. So in theory, the modification should be relatively permanent. So you can grow large batches without having to like keep the antibiotic in there, so it's safe to actually consume. So I got a question about what's your plan for the milk and egg plasmid after it's made. Mentioned distribution in some way. Right now, the main plan is I'm going to put it on Adgene because that's just the easiest. Then people can just buy it, and I don't need to deal with distribution because distributing modified organisms is tricky. But I'm also going to look into potentially distributing it myself, just as pure DNA. Because you're not allowed to ship modified organisms, so you're going to have to, like, inherently, people are going to have to modify it themselves. I might, and I mean, this is sort of the thing that I'm thinking. Like, I think at bare bones, I'll just provide the DNA, and then whoever wants it can just put it into yeast. It's not really a hard process. But depending on how involved it gets, I might provide some of the like transformation liquid, so that it's you just kind of like mix it, add it to whatever yeast you want, and it'll just do the thing. So yeah, that's kind of how I'm thinking about it, but I'm unsure. Thing one, I want to get the plasmids made and make sure they work, because if they don't work, then unfortunately it's just money on fire. But I mean, that's how bio goes. But get them, make sure they work, and then once I know that they work, definitely they'll be submitted to Adgene immediately, and then after that I'll look into setting up some other form of distribution. And making sure that I'm going to keep notes of everybody who's donated, and so once I do, or if I do set up that distribution setup, I'll make sure that anybody who's contributed will either just get it for free if you've thrown in a huge amount of money, or maybe or like free shipping or whatever. I will figure all that out as we get there, but again, this is bio, so the chances of things just failing horribly are high. Okie dokie. Let's actually get this penicillin. I think I've answered enough questions, and we've gotten plenty distracted. So let's just get this penicillin thing done. The nice thing is this should actually be really quick because they've provided almost everything we could need. Although again, it could get iffy. Okay, so we want to copy. Oh wow, this is really wonderful. They provide the translation and everything. Okay, so I just want to copy the sequence. And again, this is my first time using benchling to do this, so this could get iffy really quickly. Actually, you know what? Since we're doing this, what's on the end here? This is a terminator. Let's actually copy that as well. So let's grab the promoter. What's conscar? I don't know what this is. I'm gonna look this up. Conscar. Please don't be weird. Nope, it's weird. Okay, well, I don't know what that does. Let's see if they mention it in the paper. Con-S. No, they don't. It's just, it is a random piece of DNA that I have no idea what it does. So we're gonna grab the promoter, we're gonna grab the actual gene, and we're gonna grab the terminator as well. Oh wow, that went totally the wrong. There we go. Okay, now I've got everything. So I should be able to open up my empty backbone. Let's go to right around here. Will it jump? Or actually, yeah, okay, we gotta highlight the thing. Okie dokie. So this is the plasmid that we used last time. I believe Sal 1 is where we cut. Yeah, see, this is one thing that I don't like about benchling. With Genome Compiler, they had a better way of highlighting the restriction sites, so it just made it a little bit easier. Oh, did I have a brain fart? Oh, whoopsies. It's fine to ship GMOs, but as soon as you ship them like over borders, or with like intent to use them for like food, things get weird. But there's no restrictions on like sending DNA. Like DNA is the only DNA that's regulated produces, like infectious agents or toxins, basically. Okay, let's see if I can paste that in there. I can. Wonderful. Oh, and it stays annotated. That's awesome. Okay, cool. So that's good. So we've got the first thing. We're just gonna basically just paste exactly right at the front of here, and we're just gonna assume that that's fine. So because this is benchling, I can actually make notes whilst we work on this. So let's open up the Penicillin yeast. Actually, I'm gonna have to make a new folder for this, aren't I? Yeah. Penicillin yeast. And so let's just take some notes, so that I can see what the hell I'm doing. And this isn't really necessary because I don't think antibiotics are considered a toxin. They mean specifically like a mammalian toxin. So think like making yeast that produces like black widow venom. Like that's super illegal. That's no bueno. But if you're just making an antibiotic, especially penicillin. I mean, penicillin's toxic to some people, myself included, but it's not inherently toxic. It's an antibiotic. It's something you can buy at the store. What? Including YouTube donations. Does it really? I don't think so. But I don't know. Oh, actually, I guess maybe it will have. I'll have to check after. But yeah, if we've already, 185. 800. I think we might already actually be at the goal, but I mean, we'll keep it going because, you know, if I like, it's gonna cost more than $1,000. $1,000 is just like, I put a number there just to see if we could even approach that at all. Which apparently we're getting pretty close already. So I really appreciate that you guys are so interested in this. The companies don't really keep track of, like, they're supposed to check like what you made, but their systems are such garbage when it comes to that. Like, you can kind of make whatever, and they're like, there's no way they know. Also, there's like with bio, because things can be like the difference between like a toxin and like a structure, like just a random protein, can be very similar, but the effect is totally different. It's really hard to parse just from the code like what people are making. So yeah, it's kind of hard. Like, I mean, case in point, like there's a group of research, and people always get all freaked out about like the biohackers are gonna make a super virus or something. You don't need to worry about the biohackers. The biohackers are all broke. It's the researchers with enormous amounts of money who can really do the like freaky stuff, and also they've got clearance to just make whatever they want. So there's one group of researchers who, just to prove that they could, spent about $100,000 and had something like a dozen different synthesis companies make individual little pieces of DNA that didn't set off any other systems. But then they took all those pieces of DNA, put them together, and made horsepox, just to prove that they could. And oh boy, it worked. So yeah, that's a thing. It's kind of yeah, it's a thing. Okay, so we've cut with cell 1. Cool. I'm going to just keep my notes over here. Let's grab my empty yeast. And I'm going to end up having to re-upload the empty yeast plasmid because I didn't do non-destructive edits because I'm a turnip. But we're going with it. Okie dokie. So we grabbed, I think it was out of this. Yeah, okay. So we've grabbed the first chunk here. So yeah, con 1 scar. Yeah, it doesn't come up. Which makes me think that con 1 is a restriction enzyme. Con 1. I don't know what con 1 is. I'd bet that it's a restriction enzyme, and when they say scar, it's left over from sticking two pieces of DNA together. Okay, they made horsepox as a vaccine base, not just... Yes, fine, but they still made horsepox. The point is they made horsepox. They don't... The thing is, they don't typically make new pathogens, because that gets into, like, bioweapons territory, which is super duper illegal internationally. And also not something anybody should ever do, ever, because just fucking don't. Okay, copy. So, we've got the PCB-AB, which is, and the NPGA. So these two things we're gonna stick into, where's my empty? Okay, where are we at? Yeah, so I'm going to just paste this in right there, and we're going to see what happens. See if it actually copied properly. Oh, that's still highlighted. That's going to yell at me. Paste. Yay! Okay, I can actually just copy that way. Wonderful. Okay, so we've got the first two in. It's exciting. I'm going to close this now, so I can have a bit of a better view. Okay, so we now have our empty yeast backbone, which really is going to need to be renamed. But it's got the PCB, AB, and NPGA, which if we go back to here, just so you can see what's going on, this is the synthesis pathway. I don't know why this. I really just. Oh, this is okay. Figures. That's what we want. So yeah, this is the pathway here. So it just as a refresher, it takes these these two amino acids in this thing, and shoves it together into this long chain of junk. Basically, it's the precursor. It's called ACV. So that's what the two things that we just did. So you'll see that PCBAB and NPGA. So now if we come back over to here, you can see NPGA and PCBAB. By the looks of these actually do use different promoters, which is good. Means they'll basically be throttled on and off at different levels, although it'll probably be approximately thing. Yeah. Anyways, so we've got the precursor made at this point. So I'm gonna jump to the end because why not? Yeah, there's the mic. Okay. So I'm gonna stick the next thing in right at the end because now we've got just for organization, no other reason. We've got the promoter, piece A, terminator. Promoter, piece B, terminator. That makes our precursor. Cool. So now if we open up these other ones, we should be able, and yeah, this one has the NPGA as well, but we don't need it. That's a terminator. So we want this promoter. We want this. I might actually just be able to grab this whole thing and just be done. I just want to check. Because this one's the latest. I want to see if they've added or changed anything. But I don't see any changes. So I can grab the promoter. Nope, that's not what I wanted. Cool. So that's actually the other 3 genes that we needed. So I'm just going to give those a little copy. We're going to go over to here. We're going to just paste them in. Wow, that is a gigantic frickin' backbone. That would not transform very well at all. But that's basically it, I think. So let's just do a little checky poo here. We got NPGA, we got PCBAB, we got PCBC, we got PCLA, and we got PEN-D. So if I'm understanding this correctly, and I think I am, that means we've got this step here, we've got this step here, and we've got this step here. So in theory, we could send this to I mean, I wouldn't because it's just crazy expensive. But yeah, so from here to here to here is, I think, the bit. Yeah, so this is, man, 20,000. That's very expensive. Very, very expensive. I mean, this seemed doable until I saw how large this was. I mean, this is just kind of the nature of bio sometimes. Like, proteins that do the work can be teeny tiny, or they can just be massive. And it's very hard to tell which way it's going to go before you start designing. But if I'm looking at this right, and like I said, I think I am, I think we have the whole pathway here, which means we're basically done, I think. But yeah, 20K base pairs. So let's just, I want to say Twist is 9 cents per base pair, and there's no way they'd charge you 9 cents for something this large. Because they also have size limits. But let's just conservatively say 20,000 times 9 cents. Okay, that's, yeah, for your absolute bare minimum, cheapest, like absolute cheapest synthesis, it would cost you $1,800. But for something this big, they tend to charge more. So about double, about 36. Some places will charge 25 cents. So it's in the range of $1,800 to $5,000 just to get this plasmid made. And I mean, this is what I mean by like bio can be pay to play. Like you could, get the organisms that all these genes come from. Oh damn! Thank you, Mark A. You the boss. Floored. You guys are awesome. Freaking awesome. I love that. I love this. This whole thing has just been so much fun. Like I had to just like design some crazy ridiculous bullshit, and then we get to have it made, and then I get to share it, and we get to like do genetic engineering together. This is great. I'm thrilled. Absolutely thrilled. Okay. Anyway, so let's just wrap this up then. But yeah, so if you're gonna pay to make this, it would just cost an arm and a leg. But if you could have it made and it should just work. Like there's very little reason that I could see why this wouldn't just immediately work. Because I mean, it was designed by people and tested by people, and all we've done is essentially copy their code and just stick it into a new backbone. So I'm actually gonna rename this now. No, I don't want to close that. I just want to description. How do I do this? Like I say, I'm still pretty new to benchling. Yeah, that's what happened. Okay. I understand now. So I'm going to end up having to re-import this empty thing because this has now been modified, and I really should have done that first. But we're going to just rename this to P46815 Penicillin. And I'm going to just move this to penicillin yeast. Wonderful. So let's just close this. Yeah, and yeah, this is pretty good. I mean, I'm gonna post this on to GitHub. I'm also gonna post links, especially for this one, because it's like we've basically just copied it out of this paper. I'm definitely gonna post links to this paper and all of their stuff. Like completely, attribution goes to them. They did all the hard work. All we did was just design some stuff in benchling. But yeah, so I mean, this is basically done. We have accomplished the yeast that makes antibiotic. So if you were brave enough, you could have this synthesized, and it should just work, honestly. But yeah, so now I'm gonna since this is done, I think I'm gonna take a little bit of time to just answer some questions and hang out a little bit, and then I think we'll call it done for the day because we've accomplished the thing. If you like I said, if you've got any ideas to add to the hat, just send them to me on Twitter. Use the hashtag #Who'sGene, just so I know that that's what the tweet is about. I will take time to like just vet the ideas before they get added to the hat so that I don't end up with something too ridiculous. Yeah, that's basically the thing. In future, I am actually thinking of maybe live streaming some of the lab work on my second channel, which if you're not aware that I had a second channel, it's called The Taste Emporium. I'm also thinking of live streaming some cooking, baking-ish. I don't know about all y'all, but it's like cooking and baking is how I've been like maintaining my sanity throughout this whole ordeal. So yeah, I'm definitely thinking of doing that as well. For now, I'm just going to stick to doing these live streams. I'm thinking that I really am feeling the, do it twice a week kind of thing. Just cause, everybody's kind of locked in, and if me being a doofus on stream and like making some cool stuff is anything to like help alleviate the stress of being locked in, then I'm happy to do my part. So yeah, that'll be fun. But yeah, so I'm gonna just take some time to let me just switch this to what should we call it? Switch this to just camera view, because there's nothing else to see. I don't think. Actually, you know what? Maybe yeah. I'm gonna just switch this to camera view, really quick. Okie dokie. Now you get to just look at my face. Cool. So let's just have a little chat about just kind of whatever. Like I'll answer some questions, and if there's anything else that y'all want to say, now's the time. And then we'll call it for the day, and I will go get on with the rest of the things I need to do. Oh, I could finish adjusting the previous plasmids. That's true. I might do that. I'll take a few minutes to answer some questions, and we'll go from there. How's the lactose intolerance thing going? Like I said in the last stream, I'm making an actual update video. The short version is it's worn off, but we always expected it to. And I'm going to go into detail then. Benefit of Benchling is you can share projects. Yes, I do love that you can share projects in Benchling. I may end up making a designated Benchling account and then transferring all of my stuff over to it so that it's shareable. So that way, if y'all want to just like come in and like look at the projects, you can. I'm also just going to be sharing the plasmids on GitHub, so you can just download them, load them into Benchling, and have a look. When will your Neuron project come out? When Ben is done making the base plates, and also when the world's not on fire, because that requires going to the US, which, for those unaware, the US-Canada border is not working so good at the moment, because there's kind of an international incident, i.e. pandemic. Okay, I know I've missed a bunch of questions, so I'm going to scroll up, see what I can find. I mean, how long has this stream been going? It's 3:30. This is an hour and a half. I mean, just because we didn't spend an hour and a half looking for plasmids like we did last time, kind of sped things right along. Is it harder to work with plant or animal cells when working with genetic modification? Plant. Definitely plant. Plants are weird. Plants come with a bunch of really finicky requirements. And I've done a little bit of plant stuff, and the actual like modifying them process itself isn't super hard, but like getting things to work properly and just setting everything up and just getting them growing and interacting with them is just very, very tedious. Compared to animal cells, where you basically just like yeet some stuff into the flask, let it sit for an hour, pull it out, and you're done. I really like working with animal cells. Like I really, really enjoy doing it. I've always enjoyed doing it. The like catharsis of working with animal cells is just so much better than yeast or bacteria or plants or anything else. I like the flow. I like being in the flow hood and just like doing the thing and like pulling out stuff with the auto pipetter, and it's a lot of fun. I enjoy doing it. The modifications are easy. It's very obvious when they work. And yeah, so that's it. How do you synthesize new DNA? You pay for a company to do it, and then they literally chemically stick every individual letter together one after another, and it's tedious and dangerous and requires a machine to do it that's very expensive, and you never do it by hand. You can, but I don't know anybody who does, because that's just not fun. Okay, what else we got? Something about use what are called transform systems, put DNA in. So transformation is the process of putting DNA into the organism. So there's multiple ways to do this. There's heat shock. There's electroporation. There's other various chemical methods. It depends on the organism. My favorite is biolistic transfection. which is you literally like shoot the DNA, like you get like a tiny little, it's like a tiny little air gun that shoots tiny little particles of tungsten or gold coated in DNA. You just shoot them into the organisms. Um, it's kind of wild. I've built a gene gun before. I want to build another one just because they're not hard to build. They're just crazy, and I love them. Are any projects like Brain Cheese going to be affected by the pandemic? Yes, including Brain Cheese. Like my whole schedule for what I was filming is just on fire. So I've completely like reshuffled everything and just gotten everything working so that I can keep working. I can keep making stuff. I've been doing a lot of bio. It's hard to tell. This is my kitchen, and my my bio lab is over there. um, so yeah, I'm working through it. Nice thing is, we've always said that bio can be done in a kitchen, and it's true. It can. It doesn't require anything. Like when I was doing microbiology in university, like there was no like work in a hood. There's just like no, you sit at your bench and you do your thing, and you don't cough into your cultures, and you're fine. Were you a biological researcher? Briefly. Get further away from the camera. I can move the camera further away. Better. What is the airspeed velocity of an unladen plasmid? It depends on your on your gene gun settings. What kinds of tools of technology can I? So I think the question is like what tools, like what tech tools could make doing these things easier? It's not much, honestly. Um, a lot of the things that would make these would make like putting plasmids together easier uh, is more of a knowledge thing. It's just like knowing, like being able to like find the info in the plasmids and just stuff to work from. But it's not really a tech problem. You can't just like throw math at it to make it so. It's a lot of just it's more. Sometimes science is more art than science, Morty. Like it's the way it is. You just kind of gotta like it's from experience and knowing like okay what things work, what don't. Opinion on bioluminescent trees. Can they survive outside? Yes, actually. There's a system that a Russian group came up with that uses the bioluminescent pathway from fungi. Actually, fuck it. You know what? I'm showing you a picture. It's too cool to not just show you a picture. Fungi. Let's see if this shows up. No. Did I spell? Yes. This is the thing. This is amazing. Like this. Okay, so this is the name of the paper: Plants with Self-Sustained Luminescence. For anyone who's interested in glowing plants, this is amazing. It is the cleverest, simplest system for glowing plants that I've ever seen. So it uses the caffeic acid cycle, which is something that plants make enormous amounts. Because they use this to make lignin, which is the woody compounds in plants. So this is the caffeic acid cycle. Here, I'll make this a little bigger so y'all can see it. So yeah, caffeic acid cycle is here. And so you only need 3 genes to go from caffeic I guess it's coumaric. Yeah, so this is the caffeic acid. So it goes coumaric. No, it does actually go caffeic. So you go from caffeic acid, uses histidine synthase to just do like a boop boop, and then you get to histidine. And then from histidine, it's H3H, which turns it into 3-hydroxyhistidine, which is the actual like fuel. Like that's the actual thing that fuels the glowing reaction. And then there's one more, which it's the lux, it's the like fungal lux gene that then takes that fuel and like uses it to actually emit light. So it's only 3 genes. It's the tiniest glowing system, and it uses something that plants make a lot of. So, and the best part about this is the last step after the histidine or the hydroxyhistidine has been burned to make light, is it breaks this compound down into pyruvic acid, which is basically fuel for the plants. It is awesome. And these are the plants. Like, they really glow. Like, it's apparently visible to the naked eye. It's expressed throughout the plant very, very easily. And they actually did some stuff where they studied like how the plants react, like at different times of the day and at different points in their cycle. And it's just fantastic. So we're actually looking at essentially trying to give this a try, and see if we can like mess with the enzymes enough to get them out. So if a protein is outside of 92% similar to the source enzyme, then it's considered a different protein. And so if these ones are patented, if we can get it 92% different, if we can make the enzyme 9% different, while maintaining all the same functions, then you own it instead of the other people. So we're also trying to see if we can, because these glow green. So if we stick an RFP, like a red fluorescent protein, in fusion with the Lux protein, there's a chance that it'll pass off the green light to the RFP, because RFP takes in green light and then emits red. So if it can take in that green light and emit red, you can get a red glowing plant, which would be just freaking phenomenal, and just so damn cool. Cool. So that's that. What other we got? Saw some other questions go by. Do you have a video about how to start an amateur lab? Yes, I have 2 of them. It's how to build and how to stock a biolab. Talk about, and also my latest genetic engineering video, these bacteria are part jellyfish. Also, at the end of the video, I talk about where to buy all the reagents and chemicals, and what places to avoid, what places are good, who has like good stock, who has crap, that kind of thing. What else we got? What is the size limit for plasmids for integration into yeast? I'm not sure. I honestly don't know. I've seen some pretty big genes get shoved in and it work fine. It's really hard to say. Can you make a glowing pig and name him Piggly? No. What's his name? I know that's an Archer reference, and I'm blanking on the mad scientist's name for some reason. How did you get into this field, and was it hard to start? It definitely felt kind of obnoxious when I first got into it, just because of the sheer amount of stuff that I had to remember. Like my first biochemistry class, I mean, understandably kicked my ass. Like the professor had one rule, which is the only thing that'll be on the exam was in the slides. Problem: every lecture had 200 slides, and some of the slides had like a table of stuff on it, and anything in that table could be on the exam. He opened when we got to the final exam, he opened and like made the announcement. He's like, just so all of you know, the school has mandated that I make the exams easier because we've been sending too many students home crying at the end of the exam. So, congratulations, you get an easier exam. I was like, okay. Yeah, that was brutal. However, luckily I have a fairly decent memory, so I was fine. Also, I use a variety of memory tricks to be able to, like, I'm pretty good at cramming information into my skull, but it's a trick. Like it's not like memory is a muscle. Like it's something that you can. I mean, it's not actually a muscle, but like it works like a muscle. Like you can. It's something you can train and you can get better at. I'm gonna just leave this on a picture of these glowing plants because it's relaxing and it's wonderful. But yeah, so there's a trick called a memory palace, which basically allows you to just cram enormous amounts of stuff into your skull and be able to pull it back when you need it. You can basically, like I learned that entire biochem class 2 days before the exam, and I got an A, so it was fine. But it takes practice. Like, it was not a fun experience. There was a lot of stuff to memorize, but yeah, it's not so bad. Um, if you've got a biochem final in 2 days, learn how to use a memory palace, and then cram it all into your face because it's biochem is obnoxious. I love it, but I hate it because of the way it's taught. Like they have you like memorizing stuff that you'll never use. Like they're like you need to memorize the structure of every amino acid and their pKa and pi and just all these different values that like the reality is if I ever needed them I'd look them up. Like we have all of human like literally I have like because of cell phones, you have all of human knowledge in your pocket. Why do I need to memorize an entire table of crap that I'm only going to need once? Like, ugh, it's frustrating. How did you learn physics? I built a nuclear fusion reactor and tried not to die. Um, um, can you do insulin? Yes, actually. So a friend of mine is part of the Open Insulin um, initiative group. I don't really know. But so he's. I think I'm gonna have him on as a guest, and we're gonna be able to talk about the plasmids that they designed, and like they're making insulin. Like it's not like they're not like just fucking around. Like they've actually made some insulin, and they're working on scaling and purifying it and getting it to the point where it's usable. Because anything that gets injected into a human requires huge amounts of purification and verification to make sure it's safe. Um, so yeah, I'll have them on as a guest, and we'll do a stream like this where we can pick apart the plasmids, and um, yeah, that'd be fun. Um, no, it's not George Church. I don't know George Church personally. That would be cool, but I don't know him. Did the fusion reactor work? No, because I had to stop making it because my dad was getting uncomfortable. Like I had like thousands of dollars worth of that. I'd worked like part-time jobs and summer jobs to just like save up and buy the components on eBay. Um, and I was getting to the point where I was like starting to put them together and like get all my systems up and running, and like when I was probably like a couple months out from getting like the first little bit of radiation to come out of it, my dad was like, "Okay, you need to stop. You're freaking me out." I was like, "That's fair." Um, but the thing is, I could, if I wanted, I could build a fusion reactor in like a week and a half. If I had all the parts, they're not hard. Um, Making a good fusion reactor is a very different concept. Um, for people asking about the finger magnets, no, my magnet got taken out like a couple of weeks after that video went out because it didn't close up properly. This just happens with magnets sometimes. But one of my friends who also got a magnet the same day still has theirs. So like, it's totally arbitrary, totally variable. They're neat, but they also like the manufacturing the magnets is hard. Um, can the modified bacteria escape? Is there some mechanism like a kill switch? So biosafety is actually something that like iGEM takes very seriously, and so people have spent a fair bit of time developing like kill switch stuff. But a lot of it is you just need to be careful with how you handle your stuff. Like if you're dealing with like genetically modified organisms, like you soak the plates in bleach or autoclave them, or like you just bleach everything, and or autoclave all of the stuff that they've come in contact with, and that's usually good enough. Um, But yes, if you don't handle your shit properly, it absolutely can escape, and like various like environmental agencies take this very seriously. So it's something you gotta like be careful with. But the nice thing is most modified organisms are actually at a disadvantage. Unless you've given them something that adds like huge advantage to the organism, like they're usually at a disadvantage. Um, so they will get out competed, and then the thing will just fade. Especially like once they're out and they don't have the antibiotic selection to like keep them producing the thing. Most of the time the gene fades very quickly. Uh, um, are there more efficient ways of learning these concepts than learning them in a school environment? That depends on the person. Some people really do need a school environment to learn this stuff, whereas some people, like myself, don't. I learn very well on my own and just through just like I'll learn more if I just get my hands dirty and actually like do the thing rather than like learning about the like theory of it. Um, where so like and there's also so many resources available like with like Crash Course and that kind of thing. But for learning genetic engineering, really the only way to do it is to just do it. Like there's no like pick up a copy of the manga guide to molecular biology and like maybe like okay, so if you want to learn bio quickly, thing number one, you pick up the Noma guide to fermentation, and then you do a whole bunch of ferments because if you can ferment, you can already basically do bio. Um, then you pick up the manga guide to molecular biology, and you pick up a kit from like AminoBio or Carolina or whatever, and you work through it. And you get used to handling DNA and putting it into organisms, and then you pick a project and you design some DNA, and you have it made, and then you put it into an organism, and then you watch it fail 1,000 times. And legitimately, you'll learn so much through that process. Um, so yeah, you just do thing like grow some organisms, modify some organisms, then design something yourself. Okay, I missed another question. Oh, whoops. Saw another one go by. Car Jesus, nice name. What about combining bio and physics, like making a custom die for a laser? So it's funny that you mentioned this. I've been growing. Well, I mean, I had to throw out my culture because the pandemic hit. My lab got shut down. But before the pandemic, I was growing various fluorescent proteins so that I could extract them and then send them to Styropyro to have him try and make them into a laser. So yes, that's totally a thing, and I'm gonna try it at some point. Are you doing anything with the virus? Yes, kind of. I'm working on a video about how the testing works primarily. What else do we got? So it's the manga guide to molecular biology and the Noma guide to fermentation. Noma is one of the best restaurants in the world. They specialize in a lot of fermented goods, and they're all delicious. They're just like aggressively delicious. But yeah, they're great books. They'll teach you about doing like microbe handling, basically, and then the molecular biology will tell you more about it. Like maybe also pick up the manga guide to biochemistry or like just a biochemistry textbook or whatever. Just go through it, get the basics into your skull. How come you have interests in so many fields? Because the world is a fascinating, strange, wonderful place. Um, what else we got? How did they shut down your lab? Well, I mean, so it was shut down in the sense that like, um, you're not supposed to go anywhere. Like it's a non-essential business, so I just can't. um, uh, so there's that, and there's also the your no gatherings. So it's and they're taking that fairly seriously. So I'm trying to like do my part. I've moved the lab into my like apartment so that I can keep doing some stuff. But yeah, it's definitely inhibited my ability to do certain things. Um, would it be possible to sequence the antibodies for this virus and then grow them in a GMO microorganism? Probably, but there's going to be thousands of antibodies, and like figuring out which one is the actual like best, like most neutralizing antibody would be very hard. It's much easier to just take plasma from people who've survived, and just give it to people who are currently sick. It's called convalescent plasma. It works phenomenally well, and the FDA just approved it as well. Um, does learning a lot of disciplines make sense in our time? Yes. Specialization is for insects. The only reason I'm able to actually do all the different things that I am is because I've learned so many different things about so many different fields. Like the like being able to bounce between the physics and like biology is just chemistry and chemistry is just physics. So if you understand the physics, you can build the machines that are used in biology. If you understand the chemistry, you can understand how the biological organisms are functioning at their lowest level. And if you understand the biology itself, then you understand the like greater context of like where did the organisms come from, how do they work, how do they interact, that sort of thing. So yes, like learn as much as you can all the time, just because it's fun, not because it's like necessary. Um, I won't. I've looked at virophages. Virophages are just viruses. There's nothing really interesting about them. They don't pop. They don't wreck viruses. They just kind of hijack another virus that's already infected a thing. It's not. I don't find them that interesting. Um, what else we got? Would you ever try to find or make a new antibiotic? Technically, I've already done that. Technically, there's a really common experiment that you can do in like a micro lab where you basically take a soil sample and you mix it with water, and then you dilute that water and then plate it, and you'll get all these like little colonies of like essentially random fungi and bacteria and stuff. But sometimes you'll see like a dead zone around one of the little colonies. That colony is producing some sort of antibiotic. So one of the projects that I had to do for my microbiome class was I had to sample that one, like one of the weird ones that had a dead zone, and then grow it up and then basically purify the media to remove the bacteria and then see how it inhibited the growth of other types of bacteria. So it's very simple to do. It's very straightforward. I mean, I've got nothing better to do, so maybe I'll make a video about it. What about the spider yeast? I've spent the last 3 days working on the spider yeast, and it is unbelievably frustrating. I basically have two problems. Problem number one is getting the DNA into the yeast has been just very frustrating because I'm using, unfortunately, a weird kind of yeast that is difficult to transform. And problem number two, I tried taking the silk gene out of that plasmid and then sticking it into an E. coli plasmid so that because E. coli are much easier to transform. But I'm having issues with the PCR, so I haven't been able to actually clone the frickin' gene out. Biology is nothing if not infinitely frustrating. It's just the way that it is. Are there any publicly available resources where you can get genes that have already been synthesized someone else cheaply? AdGene. That's what AdGene is, basically. Also, the Free Genes project is pretty great. They've got a collection of different things that they've had made. Their open enzyme panel, for example, is really cool because it's just a whole bunch of different plasmids that each code for an interesting enzyme. Some of them are like restriction enzymes, for example, and you can take the plasmid, put it into E. coli, grow the E. coli, and then just pop open all of those E. coli and just like filter it out. And that crude extract can be used in place of the actual like purified enzyme. So you can like do bench work with just like E. coli extracts, and it works just fine. Will AdGene ship to a home? They prefer commercial addresses. This is sort of the one problem with AdGene. AdGene is great, but they want like a business and a commercial address. So it's best to say make friends with your local hackerspace or makerspace or community lab or whatever you've got around, or just a friend who's got a commercial property, and then register yourself as an LLC. Takes 2 seconds. So then you're like a professional entity, and then you can just order the stuff. It's a little bit of paperwork, but like then you have access to AdGene, and AdGene's amazing. Okay, we're coming up on 4 o'clock, so I'm gonna take a couple more, and then we're gonna wrap this up for the day. And then we're gonna do the next stream maybe Monday. I'm thinking like Monday and Saturday, but I might do Monday and Friday. I'm not sure, but yeah, I'm thinking I'm gonna do 2 of these a week just because it's fun and I like doing it. And like just the like working through this, it's good to just like practice, um, and just make weird, wild, wonderful things. Um, the lactose update will hopefully come next week if my experiment works. I'm waiting for some liquid to turn blue. If the if the liquid turns blue, then the video comes out next week. If the liquid does not turn blue, then it's gonna take some more time. Um, are you conducting any experiments on infectious diseases? Absolutely fucking not. That is so illegal, and you don't mess with infectious diseases unless you've got a BS4 lab. Don't do it. It's stupid. You'll fucking die. Hard no. Can you make a type of corn that will produce gluten? Actually, on my list of ideas, I have one of the ideas was make rice produce gluten. So I've already got something like that. Is there any simple lab equipment that is way more expensive than it should be? Yeah, some of it. Like PCR machines tend to charge an arm and a leg for when it's just a thing that gets hot. Or like heat blocks are like hundreds of dollars when it's just a thing that gets hot. Gel boxes can also be very expensive. I've come to really like the gel box that I'm using currently. And it was a couple $100, but like it was worth it. Some infectious diseases are BSL2. Yes, this is true. There are it depends which disease, but as a rule, I don't mess with disease. That's not something I'm interested in. It's not something I want to play with. It's not my jam at all. I work with things that are safe and are fun. I don't want to work with diseases. It's not. I'm not into it. What else we got? You could make a PCR machine DIY, but I refuse. It's just there's so many great ones that are available now. Like the Pocket PCR is like $100. Personally, I use the Pro PCR, although the Mini PCR is identical, and they're great. Highly recommend. How viable is biohacking in the EU or UK? Sorry, I just have to laugh. It is so illegal to do anything there. I've got a friend who is brilliant when it comes to designing bits of DNA. Unfortunately, he lives in Austria, so it is super illegal to actually do any of it. So he outsources. He finds other people in other countries where it's legal to do so, and just he'll pay for synthesis, and they do the lab work. It turns into these really awesome international collaborations, and it's it can be a lot of fun. You get the work done. But yeah, that's sort of the way it is. Are you a morning person or a night owl? Night owl. I don't do mornings. How do you analyze your PCR products? Do you have a gel electrophoresis equipment? Yeah, with a gel. I mean, look, I don't know the legality of every single country in the EU, but in the EU, as a rule, genetic engineering is very difficult. Like, just putting GFP into something requires a license. It's obnoxiously stupid. I will not do THC from yeast because I've got a friend who does that professionally, and he, frankly, will have done a much better job than I have. It's really hard to do. Like it's things I've learned from him is that apparently getting yeast to produce THC is very difficult. um, which country is most open to biohacking? Probably the U.S. In the U.S., you can like make genetically modified animals, and they don't care. Whereas in Canada, no working with animals, like no live animals, without a license, anyways. Um, more radio videos. Eventually, probably. I mean, I was gonna do them over the summer, but as it stands, I'm kind of locked in my house, so it's not really possible. I can't imagine the police being super amicable to me being set up in a in a park somewhere with an antenna. Um, all right, let's do one more question, and then I'm gonna wrap this up. Is it hard to source stuff in Canada? I think this is a good one. Um, kind of. Certain things are harder to come by. Most of the sellers do have international like versions. So like I buy a lot of things from a company called Gold Bio. Like they're really great for getting reagents and stuff. Um, but they have a Canadian they have a Canadian distributor. Um, the other way to do this, and I can't necessarily suggest this because it's I don't know technically the legality of it, but you can have like somebody in the states like order the thing that you need and then just like ship it up. But yeah, so it can be tricky, but I make do. Have you seen the Netflix biohacking documentary? What do you think of it? I mean, I'm in it, so um, all right, well, it's 4 o'clock, so I'm gonna wrap this up. Thanks everyone for hanging out. Thanks to all the people who donated to, get the plasmids synthesized. I'm so excited. Like I'm so happy with how much progress we made on that front. I'll look into it and hopefully I don't know, but like it's gonna take some time to get the quote back and like get everything sorted. But I will keep you guys updated on that. We're gonna do the next one of these streams on Monday, same time, same place. So yeah, I'll see you guys again on Monday. Thanks again for hanging out. This has been a lot of fun. If you've got other ideas to submit to be added to the hat, send them to me on Twitter. If you have, if you donated, I'll be keeping a list so that should the plasmids actually get made properly and everything's good, I can like get you guys some stabs. That'll be great. Um, and yeah, I will see you guys on Monday. Have a good day. Stay safe. Stay healthy. Don't get sick. All that stuff. Stay